Thank you very much. I'm delighted to be here, and I'm going to talk to you today about priming for life and how the early life nutrition really affects the microbiome gut-brain axis. So I work in a medical school, and in medicine, everything goes back to Hippocrates. And Hippocrates is thought to have said, now it could be fake news, but he's thought to have said that all diseases begin in the gut. And, that can be quite strange when I talk to a lot of my friends in areas of psychiatry and neurology and brain development. But all we have to do is really just look to our language and see how we personify our emotions in our gut. We have gut feelings, we have gut instincts, we feel gutted when we're disappointed, we make gutsy moves when we're brave, we have butterflies in our tummy when we are nervous. So, perhaps there is an underlying biology to these well-worn phrases. Now, the concept of gut-brain signaling, dates back to the times of Pavlov and Darwin, well over 200 years, and is well studied in the context of food intake. And people understand, that your gut talks to your brain to tell you to eat and when to stop eating. But more recently, in the last decade or so, there's been a greater appreciation about the role of the microbes in the gut in regulating the signaling. And that might at first seem weird, that you would want to focus on the microbes if you're interested in the brain. But really what I do need to remind you is that we are living in a microbial world. Microbes were here long before humans, and thus perhaps it's not so surprising that we have co-evolved with these friends, and these are friends with benefits. And so if we just look at some of the numbers that we have in terms of the microbes, for example, we have more than in our genes, we are more than 99% microbial. Very humbling. The weight of our gut microbes are roughly that of our brain. And if you looked at the number of cells we have more microbial cells than we do human cells. So that's really humbling as well. And with this field, well, really the two big challenges that we face, and this is what our group is really trying to understand, is how can microbes in the lumen of the gut send signals to the brain to affect emotion and behavior? What are the pathways of communication that go? And so we're slowly beginning to disentangle this, although we have still some ways to go overall. And most of this work is being carried out in animal models. And the next part of the puzzle is, how do we go from animals to humans? And that's also informing a lot of our work right now. And it's not just the microbes that are important, but it's what they're doing. So one of the big areas right now is trying to understand what these microbes are doing. And these microbes are like little factories producing all kinds of weird and wonderful chemicals that our bodies wouldn't be able to have otherwise. Among these are chemicals like short-chain fatty acids, for example. And so a lot of the focus in the research is trying to say, well, how do we get the raw materials, which is from nutrition and the diet? How do we, bring them into the factory, and then they're acted on by the workers, which are the microbes, and then to change the pathways of communication to the brain. So if we want to modulate this, we can either change the workers, but change management in workplace is difficult. And that is the same with the microbes that are there. Or we can change the nutrition, the raw materials, maybe start with something a little different, give them a heads up about what we should be doing. So that's kind of where the field is really going. And I'll share with you some of the work that we've been doing in that sphere. And perhaps the greatest example of this is really in human breast milk. Human breast milk contains the highest complexity of sugars compared to any other mammal. And evolution doesn't do that without some reason. And it was really surprising to me many years ago when I realized that these sugars, they can't even be broken down by the infant. It has no capacity. So that's not evolution working. So what's going on? Well, what's going on is that we've kind of given up that job. We've outsourced it to our microbes. And the microbes therefore break down these human milk oligosaccharides to key chemicals like sialic acid that are very important for brain development. So you can see that this coevolution of the microbiome with our nutritional environment is really important. And so this is really important throughout the lifespan and throughout early life in particular. And so there's a growing appreciation of what are the factors that influence the early life microbiome, whether it's exposure to antibiotics, to the environment, to infection, different dietary habits, various, all of these can influence both the mom's microbes and the infant's microbes. Now we get our microbes, our early microbes, kind of like a birthday present from our mothers on the day of birth. We're taught mostly to be sterile in utero. But that doesn't mean that what happens to the mom during pregnancy or even preconception, is more focus on that now, could influence the mom's microbes, both the gut microbes and the vaginal microbes that are handed off in this kind of evolutionary relay race overall. So a lot of these factors can influence the mom and then affect what type of microbes that are handed over. And then we also know nutrition during early life, whether especially in relation to mode of nutritional provision and solid food introduction, are really important aspects of shaping that early life microbiome in general. And so we've written about it in this article that's just out, which was sponsored by Ordesa, to really highlight where the different nodes within early life that the microbiome can be shaped by nutritional influences. And here we try to highlight how changes in nutrition and during this period can impact the different pathways of communication between the gut, through to the enteric nervous system, through to the immune system, up through the vagus nerve, or independent of vagus, but onto the brain to change brain function and behavior. And also another factor that we've been really interested in early life in shaping the microbiome has been mode of delivery at birth. And this goes back to work from Gloria Domingos-Bello some years ago, which showed very clearly that the microbiome of infants born by C-section is very different to that born vaginally. You can see here in blue is the cesarean and in pink the vaginally born. And that was a small study. It's been reproduced now in at least 14 or 15 different large cohort studies, most notably the TEDdy study, which highlights here that birth mode is one of the key factors, as well as mode of nutritional provision with breastfeeding also having a significant effect on the microbiome in early life. And you know, what we're beginning to see, that this microbiome is really a security blanket during early life in shaping and programming the body's systems in general. But it's not just about missing microbes, which are really important, but it's also, we need to point out that it's the blooming of pathogenic or dysbiotic microbiome overall. And so as can be seen in this work here from Trevor Lawley's group. So we're beginning to really tease this apart more and more. And we know that birth by C-section leads to a greater risk of allergy and asthma and type 1 diabetes, but we know less about it in the context of brain health, what I'm interested in. So some years ago now, and just published in the last month, work from Livia Mores, who's now at Caltech, in my group, showed that we developed an animal model of C-section. And in these animals, we could see that there were deficits in their microbiome as very early in life. In particular, we found there was reductions in bifidobacteria. And when we looked at their behavior as early as postnatal day 9, we could see changes in behavior, but all the way through at adulthood, there's this enduring effects on behavior in terms of social behavior and anxiety behavior in particular. So we wanted to go one step further and see, could we rescue this? Could we, by intervening in early life, change this. Now, mice, like some mammals, are coprophagic in that they eat each other's poo. So we took advantage of this and basically performed a fecal transplant, basically housing the vaginally born with the C-section animals to see could they basically by artificially fecally transplanting restore the effects, and indeed we could, which is really exciting. And then but that's not amenable to humans perhaps, so then we wanted to go one step further and then look at what could we put bifidobacteria back. And so we treated with a bifidobacteria breve, and we were able to reverse the effects, as well as with a prebiotic which stimulates the beneficial bacteria. So what we're beginning to see is that yes, in C-section there is deficits in the microbiome gut-brain axis, but the good news is that perhaps we can look at nutrition as an intervention strategy to try and reprogram or reverse some of these effects overall. And this is something that we're very excited about, and we feel that because C-section, although the rates are going up perhaps more than they should, it is still a medically indicated life-saving intervention, and we would like to be able to offer to moms and families the opportunity to have intervention strategies that could counter the negative impacts of C-section on the microbiome in early life in general. Okay, so that's a lot of our work, and we've been really exciting, but my neuroscience colleagues, they're a skeptical bunch. They need more evidence. How do you, to really implicate the microbiome in brain function and development? And so what we look to is other areas of science, and like engineering and biology even, where we want to get evidence that something is important. Well, you take it out and you see what happens. And so the concept of being germ-free, well, that's not, that goes back to Pasteur's time. And here you can see a germ-free man from a sci-fi magazine from the 1920s. Now, our germ-free mice, while they have their own limitations, have been very useful to allow us to answer the question, is the microbiome involved, yes or no? However, they don't really have any human translation. I mean, the exception being the boy in the bubble that Paul Simon sang about in the 1980s. And we have worked with these models over many years now, and really they've been transformative in our thinking in terms of being able to show that if you don't have microbes in this extreme situation in early life, the brain doesn't develop properly. And that's really key. And we've shown that, for example, myelination patterns, myelin is the insulation that's really important for brain conductivity, that the myelination patterns are dysregulated in the prefrontal cortex, or area of the brain involved in higher cognitive function. The regulation of adult hippocampal neurogenesis, the birth of new neurons, is out of kilter, and this is very much a process involved in higher cognitive function and pattern separation as well. A variety of changes in neurotransmitters, and more recently we've shown that synaptic plasticity doesn't function appropriately in these germ-free animals, as well as changes at the immune cells of the brain, the microglia, and at the level of the blood-brain barrier. So very exciting. And here you can see, you don't need to be a neuroscientist to see that this is a neuron from the amygdala region of the brain involved in fear learning, involved in pain processing, involved in social behavior. And you can see that it's almost like in a stress state if the microbes are not sending signals to the brain. So what does this translate to at a behavioral level? Well, at a behavioral level, it translates to deficits in fear learning, as we showed in this study, deficits in visceral pain. And visceral pain is a core symptom of adult diseases like irritable bowel syndrome, but also has very clear overlaps with infantile colic. And there may be a relationship between the microbiome gut-brain axis and colic generation, and how nutrition could be intervening in that is really important. Also, in terms of sociability, we find that mice, like humans, are quite social, but if you give them the chance to spend time with another mouse, they'll gravitate towards the mouse, but not if they're germ-free. And mice, well, like some humans, can be a bit fickle. So if you get them to spend time with a new mouse versus their everyday mouse, they'll prefer to spend time with the new mouse, but again, not if they're germ-free. And so we're finding that there are clear aspects of the social brain and social brain development that are gone awry without these microbial signals overall. In relation to germ-free being an extreme model, we've also started looking at other models, such as early life antibiotic exposure. This is a study in rats some years ago with Siobhan O'Mahony, where we showed that giving vancomycin in early life left an enduring effect on visceral pain processing. And that was kind of remarkable, showing that that even though the microbiome recovers quite quickly, as you can see on the top left here, that there are these scars of early life microbiome effects on behavior. And more recently, we've been looking at this in breaking down other antibiotic exposures that are perhaps more clinically relevant. And we looked at penicillins versus a cocktail, and we find that there are deficits in anxiety behavior, in social deficits overall. And there's something about the social brain, time and time again, that we see that makes it sensitive to what's going on. And we have examined this across the entire animal kingdom, and you start to see the data is patchy, but you start to see when you interfere with the microbiome, you can change social hierarchies and social structures. And then when you change social dynamics, you can also change the microbiome. And this is, you know, very important in this time of extreme hygiene that we have and social isolation in terms of lockdowns and what that's doing to nutrition. And so we're beginning to see that we can juxtapose what's going on in the brain with what's going on in the microbiome, and that there are critical windows in early life in particular. And we need to understand how this plays out and how this leads to changes in executive function and other higher cognitive functions overall. And also we're beginning to realize from a developmental origins of brain health perspective that the microbiome is another pillar added to both the prenatal and postnatal environment and host genetics overall. We've also been working on showing that early life trauma and early life stress, and this is data from well over a decade ago now, where we've shown that adversity in early life leads to whole body syndrome, but among this is changes in the microbiome, and this has also been shown in humans. And so the next point is, can we reverse this? And we've been working to try and do this with a variety of strategies, nutritional strategies, to reverse the impact of this early life stress on the microbiome. And here you can see some of our efforts looking at specific strains of bacteria, Lactobacillus rhamnosus, alone and in combination with prebiotics or some of its soluble mediators that are having an effect, and also looking at milk fat globule membrane and prebiotics on this. And then other approaches, nutritional approaches we've been looking at include polyunsaturated fatty acids, and more recently polyphenols, which are present in a lot of dietary substances like nuts and a variety of vegetables as well, giving a lot of color where there's a lot of color. And these flavonoids and non-flavonoids are really interesting and potentially also in early life in shaping the microbiome gut-brain axis. We've also been focusing on adolescence and the impact that nutrition, and in this case bad nutrition, can have on the microbiome and have enduring effects there. And then what we can do to kind of counter stress effects in adolescence. And this was a study together with Beatrice Pisani's group where we were able to show that supplementation with vitamin A and polyunsaturated fatty acids could reverse the effects of stress. We've also been working on antibiotic exposure during this critical period. And we feel this adolescence is an untapped area for nutritional interventions on the microbiome gut-brain axis. This brings us to the concept of psychobiotics. It's a term we've coined here in Cork, and we really need to move these animal studies into the human domain, and it means targeting the microbiome for mental health benefits. We've talked about the different approaches that we can do that. We can use probiotics, prebiotics, symbiotics. Does the bacteria have to be alive? Could we use a paraprobiotic? And then fermented foods, and we have a lot of exciting work in the fermented foods, especially in the area of kefir on this. But we feel we're at the beginning of a new revolution, a new way of thinking about shaping brain during early life and critical windows by targeting the microbiome. And we have a number of success stories coming through in our animal models, and we're moving slowly into humans on this. And this is a very exciting area. So I'll leave it at that and thank a big group of people in my team here in Cork from all over the world. And I'll leave you with the other words of Hippocrates, who's also thought to have said, let food be thy medicine, and I'll change that to let food for your microbes be medicine for your mind. Thank you very much, and I'm happy to take questions.